Yeah, thank you for inviting me. I try to elaborate on prospects of gene editing in science, and this is very difficult because we cannot really predict what will happen. So I'm trying more or less out of my imagination what could be done, what could happen, how we could manage gene editing in the next years. But of course, that has to be taken with cautiousness, with caution, and I have to add that I'm not really driving this field in my lab. So we are mainly working on stem cells. But what is the assumption I'm taking on how the technology will develop? And the first one is we know what genes do on all level of an organism, on all levels. And we in the moment, of course, don't know what will happen when we change two genes on the cell level, perhaps, but not on the broader level of a tissue, in many cases. So we will understand the human gene regulatory networks. We will understand the role of genome organization in the context of the nucleus and what that will trigger if we change it. We will predict the effects of single and multiple gene changes, and we will be able to fuse this knowledge with morphogenic effects. And the second assumption I take is that the gene editing technique is precise and predictable. So there are predictable changes in the genome upon application. There is elimination of false targets and knowledge about the effects, if we cannot eliminate off-site effects, off-site targets. And there will be routine in multiple gene editing. There will be a time and space controlled gene editing. And there will be what I call emergency editing. So if something goes wrong, you can reverse it or you can eliminate it. Are these assumptions realistic? So the first one was on we know what happens when we do something with the genome. And I will just go into one example. And we'll also try to elaborate on what artificial intelligence can play, what role it can play in this context. So this is neural crest cell. A neural crest determines a lot of our body shape, especially in the face. So the whole face is basically developed from neural crest. And we in the meanwhile know a lot about the genes controlling this manifestation of the facial structures from parallel experiments in animal models and in human in vitro stem cell-based models on the neural crest. So we know a lot about gene regulatory networks which determine how the neural crest forms facial structures. So this is shown here, maybe in a more detail. Why we do that is that many facial disorders or disorders manifesting in the face are caused by, of course, neural crest-based genetic issues. So neural crestopathies in the face are not uncommon, but are rare. And we can now depict which genes are manifesting their phenotypes in which areas of the face. So if you think that further with facial recognition algorithms in artificial intelligence, we can basically identify persons from social networks, and we can then determine the genetic background of facial features from that person. So there is a connection via artificial intelligence between facial features and the underlying genomic composition. And we can, of course, when we know that, try to modify that by gene editing. And this is just one example where we understand already quite a lot on how genes and gene networks and the genome organization in the nucleus relate to phenotypic expressions. The other assumption I was talking about was the precision of gene editing. And you probably know there are two recent approaches to improve precision. This is prime editing and base editing. They are still technologically very challenging, but there are ways into this direction. I personally believe that it will never be 100% precise. There will always be some offside effect, but as long as we can find these offside effects and determine the risk associated with these, I think it's okay. And the fewer there are, of course, the better. We can also, especially with prime editing, but also with other CRISPR-Cas methods or other editing molecules, edit multiple genes at the same time or multiple loci at the same time. Is it reversible? So there was a paper early this year, on reversibility of gene editing, which has to be specified because this gene editing is on the epigenomic level. So it edits actually the methylation of a genome, and it can, in the moment, only delete gene functions. It cannot repair gene functions. But nevertheless, it's a big step, and Jonathan Weissman from MIT was the lead author of that. It's a big step forward to silence a vast majority of genes in a reversible manner. So reversibility will be, I think, one of the future features of gene editing and also precision. In addition, with some assumed knowledge about what genes do, the tool becomes much more powerful. So what are fusion technologies? I bring that up, and I didn't find a better word for that. These are technologies which fuse somatic gene editing with embryology. So in order to modify the cells of an embryo, you don't need necessarily to do germline editing. We can avoid germline editing, basically. And that is, can be done on three parts. One is, or in two ways, in three ways. One is to edit only the cells which are supposedly contribute to the somatic compartment of the embryo, and then of course of the living organisms. You can repair genes only in those parts of the embryo which give rise to affected organs, so very similar. And you can edit genes in induced pluripotent stem cells or even in fibroblasts from a person, and then make induced pluripotent stem cells from these to generate germ cells. You just heard about this in the previous presentation. And embryos to test, for example, drug intervention to repair then gene effects. So in the embryo, somatic cells, this is actually a headline based on a research outcome from a Chinese group. Their gene editor edited a gene in the monkey, which basically replaced it with a human gene, delaying the development of the brain in this monkey, and then they found that newborn monkeys had some advantages in terms of short-term memory. This is not really only somatic gene editing, but you can use this technology basically to also just edit brain cells in an embryo. And the technology is quite known already. Just an example here. So we have technologies available already to replace organs in an animal, for example in a pig, with human organs, by using pluripotent stem cells from the human, putting them into an embryo of a pig which cannot make a certain organ, for example the kidney or the pancreas. And then if a pig will survive until birth, the organ has to be from these human cells injected into the embryo. You can do the same, actually not interspecies, but within one species. You can use a human cell which gives rise to an organ. You can silence a gene, for example, by this methylation process, which enables the human embryonic cells to make this organ, and then the organ in this embryo or in this newborn will come from the non-edited or healthy cells. So this is one avenue how we can perhaps repair genetic defects in specific organs using embryo technology. And we can, of course, use pluripotent stem cells from any person to make an embryo. This embryo can either then be directly used for in vitro fertilization or transplanted into the uterus, or it can be used for in vitro studies to see what perhaps other drugs could be used to repair the effects of the broken gene. Of course, you can also do this by making germ cells from these pluripotent stem cells, and either the gene defect can be repaired before making the germ cells, so the germ cells are basically not carrying the disease gene anymore, and can be used for in vitro fertilization-like approaches, or you can again develop an in vitro assay to see what can be done without genome editing to repair the effect. Of course, this cover picture of this book, which I showed, is not what really happens in the dish. It's more like this picture, where we have, after developing a blastoid or embryo from a pluripotent stem cell, an in vitro system with an in vitro uterus where we can study the development of the human embryo long term. And of course, we all know that long term is in the moment not allowed, really. There's a 14-day rule, but I would propose that this 14-day rule should be overruled in at least some cases to exactly study what happens with certain interventions later on in development in vitro. And then perhaps in vivo, and what else we can do is to, in this embryo, determine which cells give rise to which compartment of the human body, and then change only this cell. So this reference from Hans Spemann says we are standing and walking with parts of our body which could have been used for thinking had they developed in another part of the embryo. If we know which part we have to address to get the outcome we want, then we can probably repair specifically certain parts of the human body. So I would, in the end, come to the utility of artificial intelligence. Recently, last week actually, there was a paper out on kinematic self-replicative organisms or reconferable organisms, so xenobots based on frog stem cells. And the AI predicted how this xenobot needs to be designed in order to make new xenobots. And the scheme shows how it works. You have this xenobot in a sample of many cells, and the xenobot then moves through these cells and piles them up. And while they are piled up, they aggregate and form a new xenobot. So that's basically how this kinematic self-replication works. And it works very efficiently over several generations. The point I want to make is that you can use artificial intelligence to determine the function and the function of a cell aggregate and how this function can be brought about. So which structure is needed to do that? And that's exactly what I mean when I'm saying we want to modify the early human embryo, or also later on, to determine what shape we need to change, or what cell composition we need to change to achieve a certain effect. So, but if the technology would be precise, transient, reversible, and tissue-specific, would this be okay for gene editing in the embryo? There's a clear benefit to the individual embryo, of course, because we can repair genes which cause disease, and that if anything goes wrong with the editing, we will be able to, first of all, make it not inheritable, so germline, which is maybe wanted in some cases, but not wanted in others. It will not cause major damage to the child. It will more than just address monogenic and single nucleotide defects, and it will help to treat genetic infertility. So when in that cases would that be justified, actually? And I bring up a number of scenarios here. The first one is, of course, to support the individuals to avoid untreatable disease. This is what has been discussed, but there are other perhaps existential threats to human survival in general. So there is a massive increase in infertility rates in both sexes. So if the calculations are right, based on current trends, in 50 years, infertility will reach such a level that basically the population will not be sustainable. There's also an aging population and the need for rejuvenation of people, so extending the ability to work for a long time, which might be done by gene editing approaches. There is maybe a need to adapt the human race, or humans in general, to changes in their environment. Global warming, drought, food shortages, pandemic risks, these are all scenarios where there is a danger to the population, I would think, to express it like that. Or other approaches, space travel. You know, if we want to really travel in space through many generations, there might be genetic changes needed to enable humans to do that. And cryopreservation is another feature where gene editing can be used, for example, to express certain cryopreserving genes or proteins in every cell of the human. So these are, of course, very, very much envisioned scenarios, which are still not completely unrealistic. And when we look back in history, I mean, one of these existential threats coming out from Germany was during the Second World War, and Americans developed this nuclear bomb. And if Einstein was involved, he actually wrote the letter to Roosevelt. And now, before he died, he said, "If I would have known that the Germans fail, I would have distanced myself from it all. This was the biggest mistake of my life." So it's really difficult to bring it back when the horse is out of the stable. There was then in 1945 the Emergency Committee for Nuclear Researchers, which demanded a global government over nuclear bombs, and we all know that didn't happen. The advantages of having this technology is for a government much too big than give it out into an international body. So in summary, gene editing progress is driven and tailored by technological advances into a fusion approach, possibly circumventing germline editing. Omics, the bottom-up approach, so data are precise and widely accessible. Cell systems, a top-down approach, develop into manageable tools. So this xenobots, for example. Artificial intelligence can predict ways and outcomes, for example, in facial morphologies, and alternative technologies are developing, RNA-based, for example. So we don't need necessarily gene editing. RNA-based technologies might come up with that. And with that, I would like to thank you very much.
Thank you.